Hello students, today we will study about application of plant transformation on, or we can say it as transgenic plants. What do we mean by transgenic plant and details we will discuss in this video. So let's begin. The genetic manipulations carried out in plants for the production of transgenic plants have been described in this chapter. Okay. The ultimate goal of transgenics involved involving introduction, integration and expression of foreign genes is to improve the crops with the desired traits. Now let's know about some important list. First is resistant to the biotic stresses, that is resistant to disease caused by insects, viruses, fungi and bacteria. Resistant to abiotic stresses, herbicides, temperatures like heat, chilling, freezing, drought resistant and salinity, ozone, intense light, improvement of crop yield and quality. Example storage, long shelf life of fruits and flowers. Transgenic plants with improved nutrition. Transgenic plants as bioreactors for the manufacture of commercial products. Example proteins, vaccines, biodegradable plastics. Now let's know about the environmental stresses of plants. The different types of external stresses that influence the plant growth and development. Now we'll see. Okay, these stresses are grouped based on their characters, biotic and abiotic stresses. The biotic stresses are caused by insects, pathogens, viruses, fungi, bacteria, pathogens like viruses, fungi, bacteria, and wounds. The abiotic stress are due to herbicides, water deficiency caused by drought, temperature, salinity, ozone, and intense light. Almost all the stresses either directly or indirectly lead to the production of reactive oxygen species that create oxidative stresses to plants. This damages the cellular constituents of plants, which is associated with a reduction in plant yield. The major objective of plant biotechnology is to develop plants that are resistant to biotic and abiotic stresses. Now let's know about the resistant to biotic stresses. Genetic engineering plants has led to the development of crops with increased resistance to biotic stresses, which can be divided into three categories. First is insect resistant, second is virus resistant, and third is fungal and bacterial disease resistant. Now, first of all, we will discuss about insect resistant. Insect resistant or insect pest resistant. It is estimated that about 15% of the world's crop yield is lost to insects or pests. The damage to crops is mainly caused by insects larvae, and to some extent adult insects. The majority of the insects that damage crops belonging to some orders like Leptoptera, that is bowel worms, that is due to bowel worms, Coleoptera, that is due to beetles, Orthoptera, that is due to grasshoppers, Homoptera, that is due to aphids. Till some time ago, chemical pesticides are the only means of pest control. Scientists have been looking for alternative methods of pest control for these regions, like limitation of pesticide use. First is about 95% of the pesticide sprayed is washed away from the plant surface and accumulates in the soil. It is difficult to deliver pest to vulnerable parts of plants such as roots, stems and fruits. Chemical pesticides are not efficiently degraded in the soil, causing environmental pollution. Pesticides in general are toxic to non-target organisms, particularly humans and animals. Chemical pesticides are not efficiently degraded in soil, causing environmental pollution. Pesticides in general are toxic to non-target organisms, particularly humans and animals. It is fortunate that scientists have been able to discover new biotechnological alternatives to chemical pesticides, thereby providing insect resistant to crop plants. Transgenic plants with insect resistant transgenes have been developed. About 40 genes obtained from microorganisms of higher plants and animals have been used to provide insect resistance in crop plants. Some of the approaches for biocontrol of insects now we will discuss. First, resistance gene from microorganisms. First is resistant genes from microorganisms. Let's study. First is Bacillus thuringiensis toxin, BT. In short, BT. Bacillus thuringiensis was first discovered by Ishiwaki in 1901. Although its commercial importance was ignored until 1951, Bacillus thuringiensis is a gram-negative soil bacterium. This bacterium produces a parasporal crystalline proteinaceous toxin with insecticidal activity. The protein produced by Bacillus thuringiensis is referred to as insecticidal crystalline protein. ICPS are among the endotoxins produced by sporulating bacteria and were originally classified as gamma endotoxins to distinguish them from other classes of alpha, beta and gamma endotoxins. Now let's know about Bt toxin genes. Several strains of beta Bacillus thuringiensis producing a wide range of crystal protein, that means cry, in short cry proteins, have been identified. Further, the structure of cry genes and their corresponding toxin products has been characterized. The cry genes are classified into a large number of distinct families, about 40, designated as cry 1 to cry 40, based on their size and sequence similarities, and within each family there may be subfamilies. Thus, the total number of genes producing beta toxins, cry proteins, is more than 100. There are differences in the structure of different cry proteins. Besides certain sequence similarities, the molecular weights of cry proteins may be either large or small. There are differences in structure of different cry proteins. Besides certain sequence similarities, the molecular weights of cry protein may be either large or small. Despite the differences in cry proteins, they share a common active core of 3 domains. Now let's know about mode of action of cry proteins. Most of the Bt toxins are active against Leptopteran larvae, while some of them are specific against Dipteran and Coleopteran insects. The protoxin of cry-1 toxin group has a molecular mass of 130 kilodaltons. When this parasporal crystal is ingested by the target insect, the protoxin gets activated within its gut by a combination of alkaline pH 7.5 to 8.5 and proteolytic enzymes. This results in the conversion of prototoxin into an active toxin with a molecular weight of 68 kilodalton. The active form of toxin protein gets itself inserted into the membrane of the gut epithelial cells of the insect. This results in the formation of ion channels through which there occurs an excessive loss of cellular ATP. As a consequence, cellular metabolism ceases, insect stops feeding and becomes dehydrated and finally dies. Some workers in the recent years suggest that the Bt toxin opens cation selective pores in the membrane, leading to the inflow of cations into the cells that causes osmotic lysis and destruction of epithelial cells. That means, and finally, the death of insect larvae. Bt toxin is not toxic to humans and animals since the conversion of protoxin to toxin requires alkaline pH and specific proteases. These are absent in human and on animals. Now, let's know about Bt toxin as biopesticides. Preparations of Bt spores or isolated crystals have been used as organic biopesticides for about 50 years. This approach has not met with much success for some reasons. Let's know about these reasons. First reason is low persistence and stability of toxin on the surface of plants. Sunlight degrades toxin. Okay, so second point is the Bt toxin cannot effectively penetrate into various parts of plants, particularly roots. Cost of production is high. Now let's know about plant Bt. Let's know about Bt based genetic transformation of plants. Bacillus Bt means Bacillus thuringiensis based genetic transformation of plants. It has been possible to genetically modify plants by inserting Bt genes and provide pest resistance to these transformed plants. For an effective pest resistance, the bacterial gene in transgenic plants must possess high level expression. This obviously means that the transgene transcription should be under the effective control of promoter and terminator sequences. The early attempts to express CRY1A and CRY3A proteins under the control of CAMV 35S or Agrobacterium T DNA promoters resulted in a very low expression in tomato and potato plants. Modification of Bt CRY1A gene. Let's know about the modification of Bt CRY1A gene. The wild type transgene Bt CRY1A B was found to express at a very low levels in transgenic plants. The nucleotide sequence of this gene was modified. G plus C content altered. Several polyadenylation signals removed. ATTTA sequence deleted, etc. With appropriate sequence changes, an enormous increase in the Bt toxin product formation was observed. The transgenic Bt crops that were found to provide effective protection against insect damage were given approved for commercial planting by USA in the mid 1990s. Some biotechnological companies with their own trade names introduced several transgenic crops into the fields. Among these, only maize and cotton Bt crops are currently in use in USA. The other genetically modified plants meet with failure for various reasons. Now let's know about the advantages of transgenic plants with Bt genes. Bt genes could be expressed in all parts of the plants, including the roots and internal regions of stems and fruits. This is not possible by any chemical pesticides. Toxic proteins are produced within the plants, hence they are environmental friendly. Bt toxins are rapidly degraded in the environment. Now let's know about the problem of insect resistant Bt crops. The major limitation of Bt gene possessing transgenic plants is the environment of Bt resistant insects. The Bt toxin is a protein and the membrane receptor through which the toxin mediates its action is also a protein. It is possible that the appropriate mutations in the insect gene coding for receptor protein may reduce the toxin binding and render it ineffective. This may happen within a few generations by repeated growing of Bt crops. Several approaches are made to avoid the development of resistance in insects. Introduction of two different Bt toxins genes for the same target insect. Development of transgenic plants with two types of insect resistant genes, example Bt gene and proteinase inhibitor gene. Rotating Bt crops with non-Bt crops may also prevent the build of resistance in insect population. We'll discuss all in we'll discuss next in next video. Thank you.